Our second and final keynote speaker for this ABX meeting, Dr. John Rex. It's a real honor and a privilege to be able to introduce John Rex to you. He currently focuses on advancing novel regulatory and reimbursement paradigms for antimicrobial agents via his role as Editor-in-Chief of AMR.Solutions, a Wellcome Trust-funded newsletter focused on the latest news about antimicrobials and antibiotic resistance. He previously held roles across academia, pharmaceutical and biotech companies, and was one of the industry-based founders of New Drugs for Bad Bugs and its Drive AB project. Served as an expert in residence at the Wellcome Trust, was the founding Chief Strategy Officer for CARB-X, has been SAB Chair for the Novo REPAIR Impact Fund, and was a voting member of the US Presidential Advisory Council on Combatting Antibiotic-Resistant Bacteria during his initial 3 years of operation. He's also currently Chief Medical Officer for F2G Limited, a privately owned company developing novel antifungal agents, and an operating partner with Advent Life Sciences, a London-based venture capital group. John, thank you so much for joining us, and over to you.
Great. Thanks for having me here. Sound check, am I okay?
Perfect.
Slides okay?
Perfect, yes.
All right,
thank you.
So we're off to the races. My coordinates are on the slide. I'm happy to share this deck and also the coordinates for the newsletter are on the slide. So I want to talk, I love listening to science. You guys are doing some fabulous work, and I'm not a chemist. I will never pretend to be a chemist. I can only stand in awe of those who know how to do that. The thing that I focus on is how to turn a molecule into a drug. And there is an enormous difference between a molecule and a drug. And the stuff that you do in the lab, you need to know what it's going to feel like down the road in the clinic, because that will give you an idea about what's worth pursuing versus what is not worth pursuing. So that's really the idea of this talk, is to give you a feel for how I see a molecule 10 years down the road when you're trying to turn it into a drug. So with that, we're going to cover a variety of the topics shown here. Let's just get going. So trial design 101. Random controlled studies can be are most often randomized. Randomization is helpful, and there are a variety of ways to approach that. Anytime you can randomize, that's a strong design. It is also possible, though not preferable, to use other kinds of controls, open label series with external controls. They're generally used skeptically. It's hard to be sure the controls are well matched, and uncontrolled data is generally not useful. The paradox of antibiotics, and the reason why this is important, is that while we want new drugs for bad bugs, and while the superiority of your clever new compound is easily shown in the lab on the basis of MIC testing or such, asking for clinical data showing superiority leads to a problem. Trials actually have to be designed to avoid superiority. Instead, we have to use primarily designs having a non-inferiority statistical hypothesis. And it's easy to see this. If you have a limb-threatening infection due to MRSA, you're not going to go into a trial if I say, I'd like to randomize you to methicillin versus my new drug. Methicillin won't work, so if you get that, well, too bad. No, actually, you say, no, I actually want something that will work. And so that means that it has to be vancomycin or linezolid in the modern era. And we actually must not enroll if we think that either arm is likely to be a bad idea for you, because you could lose your leg or lose your life. And there's a YouTube explainer on that if you'd like to spend a little more time on it. You know, this why not superiority has led to lots and lots of debates over time. Because it feels like I want a better drug. You know, explain this to me. Well, you know, you can do that sort of thing in a non-life-threatening illness like migraine. You know, 12 hours of placebo, your head will hurt, but it won't kill you. Placebo is not possible in cancer, but you can always improve on 5 or 10 year survival. The thing about infections is we cure them, and you can't be more cured than cured. You know, it's just there's no up from there. But you know, this idea of non-inferiority just comes around and around. Because just the phrase itself sounds ugly. Non-inferiority. I wanted a better drug. I wanted to do studies on resistant bacteria. I understand that, but nobody wants to receive predictably ineffective therapy. And in fact, we need to stop disrespecting, as my daughters would say, it's got to stop disrespecting non-inferiority studies. They are very powerful. You say, how does new work in a multi-drug resistant infection? Well, unpack it. What you really want to know is, does new work when the pathogen is resistant to other drugs and still susceptible to new? I say a well-designed modern NI study of new versus old convincingly shows that new works as well as old when old still works. And that result doesn't evaporate because old becomes resistance. I mean, there's no cross resistance. As long as new is still active, it's susceptibility to new that matters. And by studying new while old still works, we stay ahead of the epidemic. So this is a hard, painful idea. A lot of people struggle with this. All right, regulatory rules. Agencies around the world, I'm going to talk about the US FDA and EMA. Outside of the rest of the world divides up into China, Japan, and everybody else. And China and Japan have a very different course from everybody else. Everybody else generally follows either the FDA or the EMA reasonably closely. And FDA and EMA work pretty hard to be pretty similar. So FDA approval pathways. Approval is based on substantial evidence of efficacy based on adequate and well-controlled clinical data. And efficacy is based on how a clinical endpoint grounded in how a patient feels, functions, and survives. It's not whether or not you still have bacteria in your nose, it's whether you can feel something related to that. Accelerated approval is an idea where you are approving based on a surrogate endpoint, something that predicts far distant future clinical efficacy. The most commonly used example here is HIV. HIV drugs are approved based on the surrogate marker of HIV viral load. You can't feel that, but you know that if that's high, you will do badly 20 years from now. So that's an accelerated approval. There's another idea called LPAD we're going to come to in just a second. EMA, confusingly, has labels that sound the same but don't mean the same thing, other than the first one. Approval is like the FDA definition, though it's actually not as clearly defined. There's no specific definition beyond good science. Conditional approval means your approval is based on limited initial data, and additional data are possible and are required for you to retain your marketing authorization. Finally, they have a category called exceptional circumstances, where approval is based on limited initial data and additional data are not possible. That is, you're done. And the difference between these two, most people are confused. Conditional approvals an EMA idea, accelerated approvals an FDA idea. They're not the same thing. Conditional means go generate more data. Accelerate means a surrogate measure endpoint. Antibacterials and antifungals do not ever, to my knowledge, use accelerated approval. You can get a real clinical endpoint in a few weeks or maybe a month. Accelerated has been used for TB based on sputum clearance. That has been done. I've also not seen conditional used. In general, you can generate reasonable amounts of data. It's just important. You'll sound like you know what you're talking about if you have these ideas straight in your head. But the deeper point is that approval is only the first step. You have to get everybody else to agree to pay for your new drug. Even if you get approved, limited data equals limited reimbursement. And stated differently, much of the problem is not regulatory. The agencies are the first to articulate the challenge, but everybody else piles in later on. So when I hear somebody say, oh, we need more regulatory flexibility, I can tell you there's already a lot of regulatory flexibility, and the regulators aren't the ones who are really standing in the way. It's the need to generate data that convince people that what you're doing, what your drug does something, that's the hard thing. So the standard pathway. So based on all these ideas, what is emerging for new traditional antibiotics, and something that sort of that kills bacteria straight out, like one of the compounds we're just being discussed that inhibits topo. You do one good RCT versus a good comparator in a usual drug resistance setting where both drugs are predicted to be active. You do it in one of the major indications, and you clearly show safety and efficacy based on a nice, clean data set. You can do a CUTI study in 18 months. Done. It's, straightforward. You also will do something to pick up some data in highly resistant pathogens. It's going to be a salvage kind of a design. You might randomize if you can, if there is a best alternative therapy. It could be open label if the N is just too small for that. And an example of that, this has been replicated by several compounds. So plasmamycin, they did exactly what I just described. They did 1X CUTI study, and they did a salvage study. Now, there's another idea that gets woven in here, which is that is of a pathogen specific. I want to study just one bug. I want to study Pseudomonas. And my agent only works for Pseudomonas. Okay, so that's the idea here. And we talked about this some years ago and called it a tier C pathway. That is a rare pathogen only MDR pathogens or only rare diseases, small trial programs, just barely or not powered. And the idea we came up with at the time was you could think of trial programs as having 4 possible levels of data. On the far left, trial tier A, you can do 2 big phase 3 studies. You can generate a lot of clinical efficacy data. On the far right, you're studying anthrax, where there aren't any human cases, you hope. And what you're going to need to do in tier D is get your approval based on purely animal data and some phase 1 PK data in human beings to prove the drug actually gets the PK that looks like it ought to be required. And then we said in between, there's a B and a C, and a B is a cut back version of A. It's one good phase 3 study and some other stuff. So that would be like the plazomicin program. But then tier C was one where all you could really do realistically was some small-ish studies that were focused on a pathogen. And this concept of tier C, you know, we've batted it around a lot. And the bottom line is that small programs may or may not be okay. The EMA says they'll take an underpowered RCT, a teeny tiny RCT, just do some randomization, but I've actually not seen this happen. And when you've got a really tiny program, there's a problem with heterogeneity. Even if you're doing 20 versus 20, every patient is 5% of your endpoint, and it only takes 1 or 2 people moving because they're slightly heterogeneous to, you know, make the data look bad. And the FDA really won't go there. Their rule is substantial evidence of efficacy based on adequate and well-controlled clinical efficacy data. Tattoo that on the inside of your eyeglasses. I mean, that's the message for FDA. And it's not just a regulatory hurdle. The strength of evidence, if you don't have acceptable data, will be frustrating. So my advice in this space is don't do it. And if you have to do it, don't do it. And if you really have to do it, find an expert and try to do some other path. And ultimately, you can possibly do this. Entasis reasonably did a challenging but impressive study against Acinetobacter, where they demonstrated that their new compound works better than old compounds. But it's a tricky idea. And the existence of their new compound will now actually make the next drug even harder to develop. It's fascinating how it is. We finally get a good tool that makes the next tool harder to develop. Okay, LPAD. This is a US only idea. LPAD, actually it ought to be LPAD, limited population antibacterial and antifungal drug. It started off as an idea that would allow FDA to approve with a more limited data package. But it got approved in 2016 as part of a thing called the 21st Century Cures Act. But while it was working its way forward, the FDA actually implemented the equivalent of tier B. And really LPAD is sort of a mashup of tier B and tier C, more tier B than it is tier C. And FDA actually said, you know, we can do tier, we've got this flexibility, we can do that. And it actually in about 2012, it became possible to get antibacterial approved with 1 RCT rather than 2. So the FDA did that. But LPAD is still a useful idea because it gives us 2 gifts. The first is the very name is a clear reminder that patients and physicians make different risk-benefit decisions when options are limited. LPAD defines such settings, and it helps the regulators and the community see that a small data set that demonstrates a clear signal, even if it's not the usual size of data. No, I don't have 300 patients for the safety database. No, I don't have quite the X or the Y or the Z, but you can see that it works. And there's this very sharply defined group of people who need this compound. And it also is a way to mark LPAD approved drugs as different. There's some labeling language. You get the limited population, you get that in capital letters in several places in your label. And so combined with robust stewardship programs and CDC's ongoing surveillance, we can be comfortable that an LPAD agent would be used wisely. So that's the other approval pathway in the United States. Approval, accelerated approval, and LPAD. All right. I get asked about this one a lot. Agents that augment, virulence modifiers and so forth. You've got something that, you know, a virulence modifier or an antitoxin, okay? And the notion here is you're going to add this on to something else. And anytime you get in the land of add-on, where you're going to have an active base agent and you're going to put something with it that improves the outcome with the base agent, you're in this space. And there's some distinctive hurdles here. The first is the base therapy needs to work. You know, if the add-on depends on the base therapy getting you going, it's obvious you don't need that. It doesn't solve existing resistance problems. Second is you may have an issue with selecting dose of your thing. If you don't have an MIC, it's harder to apply PK/PD based dose selection. That means it's impossible. I've seen people come up with clever approaches here. It's just harder. You might need a novel endpoint to show value. We actually once debated a product where the purpose of the product was to prevent the development of resistance. It sounded like a great idea. I could not figure out how to turn it into an endpoint we could work with. And finally, there's the superiority problem. You're going to have to show that when you add the new thing to the old thing, it's actually better than the old thing. And that's the numbers game problem that we'll talk about on the next slide. Before I leave this, though, read the papers at the bottom of this slide if you're thinking about doing this kind of work. So here's the math problem. Ultimately, you're going to have to show that new plus standard of care is better than standard of care. I mean, nobody's going to care if new plus standard of care is the same as standard of care. I mean, why would I add it on if it doesn't get me something? And it's going to have to be based in something that a human being, can perceive. Okay, feels, functions, survives. This reduces to a math problem. Typical active drug, CUTI, anything, 85% response. See about a 15% failure. Of those 15%, loosely, some of them are out of your hands. Underlying disease, other factors, nothing is perfect. Let's just say 5% are out of your hands. That means you got 10% where your add-on can improve things. You got this itty bitty window where you're going to make a bump. And if you say, well, I want to go from 90%, so I got the 85% who will respond, I got the 5% who can't respond, so I've got this 10% left. I want to go from 90 to 95. My N is 1,240, 620 patients per arm. That's a huge study. And I want to go from 90 to 92.5. Well, that's an even bigger study. And quite honestly, would you care? And so it really is it's a challenge here. So my feeling on this is there are settings where you might achieve it. So truly resistant pathogens are reasonably frequent, and the comparator arm isn't 85%, it's 60%. Okay, there you got more room to work with. Endocarditis, maybe you could do something with more rapid bloodstream clearance that might actually predict a measurable clinical effect. Chronic infections may offer a little more scope here. Different endpoints, we've debated this. I just mentioned the idea of showing a reduced rate of onset. A community benefit, we've talked about it, had an afternoon or workshop about this, could not come up with a great way to deal with this one. And I'll say it again, it's not a regulatory problem. The agencies are going to point it out. They're going to say, explain to us why we want to use this. And so my bottom line is, if you're going to go down this route, whatever you're developing had better be amazing. Otherwise, you are wasting your time. It's a nice science project, get you a paper, but it's not a drug. Okay, getting towards the end here. New reimbursement paradigms. Hopefully you followed the literature about push and pull. And this is an idea this pair of ideas that developed over the last 10 years, in which we think about how we can incentivize the development of new drugs. And push incentives are the kinds of things that help you get your little company going, grants. And there's a fair bit of this money out there, money for discovery to phase 1 in CARB-X and Novo REPAIR. There's the $1 billion AMR Action Fund that is going to fund phase 2 and phase 3. And then we need this idea called pull incentives, which are paid on successful approval. That is a payment to the innovator for the availability of the compound, even if it's not going to get used. And if this idea is new to you, there's a lot to read. Start with the YouTube explainer on this slide. Oh, I didn't mean to play that. The idea of a pull incentive is that it would equalize the economics. And the concept is that it gets paid on approval. It's independent of actual use. And we think about this in the sort of the fire extinguisher model of antibiotics, which is, of course, the logo on the slide. Think about the fire department. We actually pay for the fire department every year, even though we hope actually not to have our house burned down, right? But you don't pay the firefighters per fire, because that actually leads to bad behavior historically. You pay to have the service available. And so a new antibiotic is like that. Antibiotics are the fire extinguishers of medicine. And what you want to do is create the antibiotic and then pay to have it available. And the most advanced versions of this is the one in the UK called, gets the shorthand for it is the UK Netflix model. And the UK is in the process of contracting for, we hope, about 10 million pounds per year for 10 years for a good antibiotic. The UK is saying implicitly the availability to have a really good antibiotic in our pharmacy, whether we use it or not, is worth 10 million pounds a year, 100 million pounds over 10 years to us, to the UK. And if you scale that up by the UK as 3% of the G20, it's about, they're saying it's worth about $4 billion to the economies of the world, which is about the right number. Economic research says that you do need for this incentive to be in that range for it to cause the whole ecosystem of all the people who want to do this work to come into being. A lot of discussion about that, lots of links at the bottom. And the challenge these days is to engage and extend. And it's really exciting. The UK is the most advanced. The UK has a thing called the Pasteur Act. The EMA has recently announced that they're going to get into the game and work on this problem. And they're actually spinning up some working groups to actually do the research so that the EU can figure out how best to do it for the EU. And at the recent G7 finance ministers meeting, the G7 finance ministers all signed on to saying that we need to do something like this. And we don't know what it's going to look like, but we all need to find a way to row the boat in the same direction. The UK pilot scheme is going to set the pace for lots of stuff. And importantly, they have a point scoring scheme that tells you what they value. They've laid it out. Here it is. You can look at your project today and say to myself, okay, what do I think I could get in terms of points? Really helpful. I'll let you read about this. The Pasteur Act, everything is going to have a very similar feel to this. It's just, it can't be that much different. All right, so in summary, a standard non-inferiority to design randomized trial and a standard indication is the most reliable path to approval. The FDA and EMA are similar but not identical. One global program is possible with CARE. Pathogen-specific approval is not yet a well-established pathway. The Entasis example is a good start, but they actually did they took advantage of the fact that their comparator arm actually was worse and there was more blue sky for them to work with. And that means the next drug is going to be harder. Agents that augment are very, 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 very hard to develop. And new reimbursement paradigms will strongly favor innovation, but the product still has to be developable. So that's it. Thank you very much for your attention. And I am delighted that all of you are putting your time and your effort into thinking about developing new antibiotics for us, for our children, and for our children's children. Stopping now.